bit of extra time right here, so that's exciting because I really jammed a lot into this lecture. Corey did cover some of it already, so that's nice. But graceful aging, right? We want that longevity, and we want to feel good in our bodies as we age. So this lovely lecture is about keeping your body and brain protected, healthy, and strong for the next 50 years or more. And our learning objectives today are to define what aging is, and we'll review the theories, identify the key SNPs related to aging that we utilize, develop an understanding of how aging affects organ systems, learn how the SNPs affect the areas of aging, and develop an outline for how to assess aging and create a treatment influenced by those single nucleotide polymorphisms and always clinical symptoms. So here's our presentation overview. First, what is aging? Relevant single nucleotide polymorphisms. What are the applications of the single nucleotide polymorphism research? Personalized aging medicine, and finally, treatment strategy. So, aging, right? It's a natural life process accelerated by environmental exposures, toxic burden, and physiological stress. There are actually quite a few theories. I didn't realize this until I started doing a little bit of research, but there are multiple theories of what's going on with aging. And one of the big ones is that it's, an issue of growth or hyperfunction on a molecular level. There's this progressive accumulation of molecular damage that's caused by environmental and metabolically generated free radicals that can cause these spontaneous errors in our biochemistry. And these can lead to hypertrophic changes, hyperplastic pathologies, and this sort of relates to the theory of persistent developmental programs. So your face when you realize how much you've aged. I love Kristen Wiig. I think she's a hoot, and this cracked me up. So this woman started working out in her 50s. Her name is Ernestine Shepard. This woman is much of our current geriatric population, right? She's happy, she's functioning, but utilizes a walker and, maybe has diabetes, but yet they're both 74 years old. So yes, genetics plays a big role, but also epigenetics, right? Ernestine Shepard didn't start lifting till she was in her 50s, and she looks amazing. So we all can take a huge role in controlling our destiny when it comes to aging, and polymorphisms, single nucleotide polymorphisms really give us a leg up in terms of identifying our potential risk factors and treating them as a preventative measure instead of waiting until we're already in trouble. So we're going to talk about hormone health, cognitive function, cardiovascular function, mood changes associated with aging, and then bone health. Hormones and aging. So this is definitely not all of the hormones. When I initially made this lecture, I realized that this whole day could have been on this lecture. There are just so many different things that we can look at in terms of aging. The three that I chose to focus on, and we talked a little bit about them with Dr. Edwards, were catecholamine-methyltransferase, CYP1A1, and monoamine oxidase A we're going to look at. And then COMT deactivates catecholamines, converts catechol-estrogens to methoxyestrogen, and also is responsible for xenoestrogens. So we'll look at that one. CYP1A1 is a cytochrome P450 enzyme responsible for drug and xenobiotic metabolism, and then MAOA is involved in catecholamine and serotonin metabolism, and histamine, but that's MAOB, excuse me. COMT. COMT is a catecholaminomethyltransferase enzyme, and this is a big one in terms of adding a methyl group to sort of break down our catechoestrogens and our catecholamines. It requires SAMe, obviously, because as we'll talk about in our methylation lecture later, SAMe is one of our greatest one-carbon donors, or our methyl group donors. So it requires SAMe, and it also requires magnesium to function properly. And when we're looking at whether we have an upregulation or downregulation in catechol-aminomethyltransferase function, we really want to assess, okay, well, wait, if we're upregulating, we're going to be running through that magnesium and those SAM-E cofactors, which are going to be B12, B6, folate. We're going to be running through them more rapidly, and we really need to support our patient there. This is redundant, that line there. So low COMT, which Corey already talked about, this is one of the places that SNPs get really confusing. So COMT can be reported as MET-MET, which is looking at the amino acids, AA, looking at the base pairs, or this could be reported as SS because it's slow. So when you're looking at these research studies, it takes a lot of work to determine, okay, wait, what are they talking about here? And that's why we do the work we do, right? So we can discern what's being said for you. But so interestingly, a MET-MET is going to slow down the breakdown of our catechol estrogens. But counterintuitively, estrogen also slows down the COMT enzyme. So we have research, and this is not research that has huge meta-analyses behind it. So I just want to put that out there. But there's research showing that estrogen slows down COMT. So if we're looking at an estrogen-dominant patient, these are factors you really want to take into account. It's not just what the single nucleotide polymorphism is, but we need to identify the entire presentation of our patient when we determine how we treat. BAL-BAL or GG or FF for fast, that's going to be more rapid and result in lower catechol estrogens because they're going to break down more rapidly, and typically we associate that with a more positive outcome in terms of risk of cancer because, as Corey was stating, those hydroxyestrogens tend to be more carcinogenic as compared to the methoxyestrogens, that we're going to be looking at in a second. And then this is just a small study of perimenopausal women that found after a single oral dose of estrogen, when you looked at the serum levels in these women 2 hours later, based on their catechol-aminomethyltransferase presentation, the level of estrogen in their blood was going to be variable. So that's just sort of proving the point there. I feel like I talked about this already, but here's an image. I feel like this is helpful in terms of determining what we were talking about, where things are breaking down, right? We've got our catechol estrogens here. Corey was talking about CYP1A1, and I'll get more into depth on that. But we've got here E1 and E2, and we require CYP1A1. This is the most research in terms of breaking it down into 2-hydroxyestrogen. And then CYP1B1 is going to be more responsible for the 4-hydroxyestrogen breakdown. Both are required and both utilize COMT to be converted to the less carcinogenic form of methoxyestrogen. But if our COMT is slow, we are going to end up, it's possible that things are pushed more into this quinone pathway. And this has to do with redox cycling. And if we are pushing in this pathway more intensely, we utilize our glutathione to balance things out. And as our glutathione is depleted, we end up with DNA damage. And these DNA adducts are what push us towards a more carcinogenic state. I don't even have that in my notes here. So, okay. Cytochrome P450 pathways. So the cytochrome P450 pathways are a group of super enzymes. There are many. We know that there are more than 57. We metabolize a vast array of compounds. So drugs and toxins, endogenous molecules are reliant on our cytochrome pathways to be broken down. CYPs account for 75% of all drug metabolism. We have a big PGX panel at Kashi that really is more emphasizing that information, whereas in terms of aging, we really focus on the CYP1A1. But these cytochromes are mostly found in the liver, and then it's a handful of them that does most of the drug metabolism. And each cytochrome has multiple versions. They sort of relate to different parts of our liver detoxification pathways, which I'll delve into in a little bit. But for example, CYP2D6 has over 70 different alleles. So, right, we can look at these different alleles and how they're going to affect the different enzymes. And then, in terms of metabolizing drugs and hormones, multiple cytochromes are necessary to break them down. It's not just one, right? So, when we're looking at estradiol here to break it down, you already saw this in the previous page, but we've got CYP1B1, 1A1, 1A2, 3A, and 3A, all right here. And that's only part of the breakdown process. All right. So CYP1B1, obviously part of the family. We already talked about how it's required to convert our estrogen into the less carcinogenic of the two catechol estrogens, 2-hydroxyestrogen. Right there. Okay. So it's most pronounced for the catalytic activity of 2-hydroxyestrogen. It's found that the 2-hydroxyestrogen can inhibit cell growth and proliferation, but it's also found that it can damage DNA and generate free radicals. So CYP1A1 can push our production of the 2-hydroxyestrogen, but we can still go down that pathway to the quinones and the quinone pathway where we're ending up causing that DNA damage. Rapid COMT is going to increase the metabolism of 2-hydroxyestrogen, and therefore, as we see it, we saw in that image, it gets pushed towards being broken down, whereas the 4-hydroxyestrogen is more commonly going to be pushed into the quinone pathways, where we're more likely to end up in this oxidative stress state, where we can see those DNA adducts produced, which cause damage and lead to carcinogenesis. And the point is, we want to look at these puppies together, right? Because the enzyme activity of one and the other can sort of balance each other out, or they can both be upregulated. So, like Corey had talked about, you can't look at just one single nucleotide polymorphism. And as we learn more, this is going to be more straightforward. But at this point, we're still cutting edge, right? We're learning every day. And you have to look at your clinical presentation of your patient and use different lab tests in combination, which we'll talk about more when we get to the labs matches made in heaven. So this is one of our diagrams that we utilize to demonstrate COMT's effects on estrogen and neurotransmitter function as well, which we'll talk about more as I jump into the neurotransmitter aspect and mood in aging, but those are available to you. MAOA is a monoamine oxidase enzyme, so it's breaking down monoamine oxidases such as our amines, our serotonin, our histamine is MAOB, and then catecholamines like dopamine, norepinephrine, and epinephrine. Studies on progesterone's influence on MAOA are just not abundant, and what we have is conflicting. But the studies on estrogen show that estrogen also, not only does it have that inhibitory effect on COMT, but it also inhibits monoamine oxidase A. So we want to look at those guys in combination, and we really want to consider what's going on with hormones when we're looking at these polymorphisms. MAOA requires magnesium, B6, and vitamin C for appropriate function. So you want to always be assessing your patients. I mean, we all know, functional medicine, diet is integral to your patient's health and digestive function. You can eat well and not absorb, so we've got to work on that. Depending on monoamine oxidase allelic presentation, estrogen could be beneficial for regulating the rate of bioamine degradation. So the status of this polymorphism can influence your decision whether you want to use bioidentical hormones with a patient or not. And this is just a fun little image. MAOA and COMT are both referred to as warrior genes, but it just is showing how the gene encodes for the protein, which then affects our neurotransmitters. All right, cognitive function. Well, gosh, we all know about the joy of aging in our brain and memory and all those goodies. I work with primarily children with Down syndrome, and so Alzheimer's risk is nearly 100% in that population. So this is close to my heart, looking at these polymorphisms that can affect our cognitive function. I'm going to talk a little bit about apolipoprotein E, which Corey already discussed. We'll talk about MTHFR. There's some really new, interesting meta-analyses out on that in combination with ApoE, nitric oxide synthase 3 or endothelial nitric oxide, and DRD2. So apolipoprotein E, I put a bunch of information on here. I am not going to read this slide, but I'm going to just talk to you about it, and this will be there for you to read on your own. But basically, ApoE, excuse me, is a protein, or makes a protein that becomes a lipoprotein, and it's going to affect our VLDL and HDL specifically, and depending on your polymorphism, it will affect your LDL levels. ApoE3 is the most common. ApoE4 is the strongest risk factor for late onset or sporadic Alzheimer's. E2 has a really low commonality, so we see that in 5 to 10% of the population. E3 is 65 to 70% of the population, and E4 is 15 to 20% of the population. But this is a little bit different than most of the SNPs we're looking at, because to determine ApoE status, you're not just looking at one location, you're looking at the combination of two. We don't really have much information about ApoE1. What you find that we dictate or what we determine in our genetic testing is most frequently the ApoE2, E4, ApoE2, E2, E2, E3, 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 and E3, E4, based on the risk associated with the combination. It's either going to be E2, E3, or E4. So ApoE4 likely causes neurodegeneration secondary to its structure. And this goes back to Corey's really first lecture on the structure of proteins and the amino acids and how they can develop, how they can influence the structure. And we have this switch in an amino acid which changes the structure of apolipoprotein 4 so that the receptor for LDL is blocked. And that's why we see higher serum LDL, because it's not binding to the ApoE4 lipoprotein. So this altered structure, we end up seeing, mitochondrial dysfunction, cytoskeletal alterations, tau phosphorylation, impaired synaptogenesis, increased amyloid beta protein, lysosomal leakage. So you can see, based on those things, why there would be an increased risk for cognitive dysfunction or Alzheimer's with this protein. And this is just an image showing you the difference in the structures between ApoE3 and E4. You see, it's hard to see here. Okay, so this cytosine in ApoE3 is an arginine in ApoE4. So as you can see, it sort of shifts over, and this closes down and blocks the receptor. And what you see in this image here is that stress and injury can affect the cell, and then as ApoE is synthesized, ApoE4 breaks off these neurotoxic fragments that lead to this mitochondrial dysfunction and cytoskeletal disruption. And that's why ApoE is such a fun one to identify in a patient, because it gives you a great advantage in terms of cardiovascular health and cognitive function. Or I think it's fun. So I'll briefly just talk about endothelial nitric oxide synthase. We've got a few different studies on it. This meta-analysis shows that the polymorphism is associated with an increased risk of Alzheimer's disease. DRD2, we don't think of DRD2 so much as cognitive, although there is a relationship. This particular study talks about when we look at populations that exercise versus populations that don't exercise, the folks who are exercising, as they age, there's significantly less significantly less loss of dopamine receptors. So this is just a push to emphasize for your patients, lifestyle matters. Eating well, exercising, this is the foundation of everything. MTHFR, so this is the 2017 meta-analysis I was talking about that's really exciting. We have so much research on MTHFR and so much of it is conflicting. This is a great meta-analysis that supports MTHFR C677T polymorphism it's associated with an increased risk in Alzheimer's disease. And I think probably many of us would have said, yeah, that probably is associated, but it's lovely to have this piece of information available to us so you can show your patients, hey, you have MTHFR and you have ApoE4, we really need to be careful. I mean, this is just such an amazing way to inspire your patient to change. And some patients will just listen, but a lot of patients need that extra push. Okay, aging in terms of cardiovascular health. I'm going to talk about ApoE like a little bit more, but a lot of the same things come up, MTHFR, nitric oxide synthase, and then two of the other ones that I'm going to talk about are 9p21 and angiotensinogen. These are the ones I should have noted. So the ones that I highlighted, this is when you're looking at our DYD panel, the graceful aging is the highlighted ones. The ones that aren't highlighted are not in the graceful aging, but that doesn't mean they're in that DYD report. So this is why it's important to have this information so you can pull it all together and see how you can help your patient most effectively. ApoE we talked about already, MTHFR, folate metabolism, hyperhomocysteine, increased risk of cardiovascular disease. Nitric oxide synthase is necessary for vasodilation and appropriate circulation. I remember in medical school, like I just couldn't remember what nitric oxide did until one of my tutors told me. Just think, when there's erectile dysfunction, you need to give support for nitric oxide so that we can increase viability, if you will, or not viability, you know what I'm talking about, virility, excuse me. ApoE2. So ApoE2 increases atherogenic lipoprotein levels. So we really think mostly of ApoE4 as being the really risky lipoprotein polymorphism to have, but ApoE2 does have some research on increasing atherogenic lipoprotein levels. Binds poorly to LDL, so similar to that ApoE4 structure issue I was showing you, we can end up with increased serum LDL, which can lead to cardiovascular problems. This we sort of already talked about, and you can read on your own, but basically increased plasma triglycerides and lipoprotein records. ApoE can be gender dependent, and I just really added this little image because I thought it was sort of fun to look at how ApoE ends up causing the peroxidation of lipids, platelet aggregation, and it can modulate our lipoprotein metabolism. I'm way off here in terms of my... Okay, so endothelial nitric oxide increases ischemic heart disease. We sort of already know that, but as we age, it is affected and is therefore increasing the ischemic heart disease. And when we look at these in combination, we want to make sure and assess that endothelial nitric oxide specifically. Increased risk, let's see. I think basically you guys understand that, but there's increased MI risk with this as well, atherosclerotic disease, DVT risk after surgery, and insulin resistance and stroke. Oh, I don't know where I am, but I better start moving quickly. Okay, MTHFR. Our results support MTHFR 677T for the Asian maternal population for congenital heart disease, and then A1298C was seen as a risk factor in the Caucasian pediatric population. So this is just an example of how each RSID number is going to be related to different outcomes, and its cultural and ethnic background is going to affect what that polymorphism means for someone. This one I take into account because I see a lot of kiddos with congenital heart defects. So 9p21 is a really biggie because this is one of the first and most well-established genetic markers for coronary artery disease. So it's a really good one to know. We have strong evidence that this is a major risk factor. Let's see, one allele increases the risk by 15 to 35% of coronary artery disease, and if you have both or you're homozygous, there's a 30 to 70% increase in risk. So having a healthy life, eating a Mediterranean diet or a plant-heavy diet is going to be more advantageous, and knowing this information helps to motivate your patient. Angiotensinogen has to do with our renin-angiotensin-aldosterone system. We all sort of think about that, obviously, in terms of blood pressure. This particular study talks about having the polymorphism could be a prediction marker for risk of myocardial infarctions. This one's particularly in Asian population. Let's see, no associations were detected with the risk of MI in Caucasians in this particular study. Mood. So redundant, right? These enzymes play roles in so many different ways. COMP's coming up again, MTHFR, MAOA, ESR1. So I need to be very clear that the RSID on this ESR1 is not the same as what we test. So I just want to point that out. But it is a very important piece of information. And then DRD2 is also going to impact reward processing. And there's a lot of relationship between our hormonal fluctuations and enzyme activity. So this is just a quick picture showing you, here are some of our neurotransmitters and phonoamines and the breakdown involving MAOA and COMT. This is just delving a little bit more into depth though. So hormone fluctuations are affected by COMT and MAO activity. So ESR1, as it affects estrogen levels, can cause downstream effects on COMT and MAOA. MAOA, there's an incidence or severity of depressive symptoms that's greatly increased in women during menopause due to hormonal changes. So once again, we're just sort of reiterating that relationship between hormones and enzyme function. MTHFR, as we have hyperhomocystinemia and less, or actually not hyperhomocystinemia, that's the issue here, but the carbon donors, the availability of methyl groups, if we have a change in the availability of the methyl groups, that is going to affect catecholamine synthesis downstream, right? You guys remember that COMT uses the transfers the methyl group. And then ESR1, so estrogen receptors, that is the means of how estrogens exert their hormonal influence in the body. So estrogen promotes epinergic and norepinergic neurotransmission, and as that shifts, mood will shift by its relationship with neurotransmitter breakdown. DRD2 is a dopamine receptor. We talked about this in terms of weight, because when we're thinking of that sort of satiation, that mentality around the way we eat food, that addictive personality in terms of food addiction, this also can relate to mood. So, one meta-analysis with 2,100 cases and 32,000 controls made the association that DRD2 is related, the polymorphisms are related to mood disorders. And, I don't have an image in here. There's one image that I don't have in there that just shows the relationship between our endocrine system, our nervous system, and our immune system, and how our hormones and our neurotransmitters really interact with one another, and we need to consider them when we're looking at all of these different factors. So bone building, the big ones I'm going to look at are the ESRs and WNT16. But we'll also discuss some of the vitamin D receptors and the vitamin D, the enzymes influencing vitamin D status. So here's a lovely little picture of how estrogen receptors are all over the place, right? They're not just going to affect our reproductive organs, but they're also going to affect our central nervous system, our bone health, and that's really what we're focusing on here. We want to clarify that estrogens induce cellular changes through the binding of one of the specific receptors. So looking at these polymorphisms gives us some idea of the risk of fracture. ESR1 is a susceptibility gene for fractures. So XBEL, this is another way that we can sort of make it really confusing to know which risk allele you're talking about, because XBEL is a combination of alleles that determines fracture risk by mechanisms independent of bone mineral density. So knowing ESR1 status can help you. ESR2 also plays into this as well as an estrogen receptor. And there's significant research on the relationship between ESR or estrogen receptors overall and the ovaries' secretion of hormones affecting calcium and vitamin D balance. Okay, WNT16 is a really complicated one in terms of how it relates to things. The underlying etiology of how WNT16 affects bone health is complicated, but there's plenty of research showing that biochemistry and the change in our bone health causes increased risk in fracture. And then, here, hold on a second. So there can be differentially expressed in normal tissues. And then they're mostly expressed kidney, placenta, brain, hearts. But studies show that WNT16 is strongly associated with cortical bone thickness and fracture susceptibility in humans. So this is just a little image showing how, it's sort of hard to see, but the cortical bone is affected by those WNTs here. Okay. So this particular slide delves into vitamin D receptors, and so NADS and DHCR7, this is dehydroxycholesterol dehydroxycholesterolase, and the relationship between these two is linkage disequilibrium, which I thought we were going to cover a little bit, but basically the location of these two genes, the location of them is in proximity enough that they affect one another. So that's sort of why it plays in. And then NADSIN also relates to the production of nicotinamide adenine dinucleotase, which then feeds back on dehydroxycholesterase. So I'm not going to bother getting into this one too much. You can read that on your own. But here we look at the action of these particular enzymes. So as the sun comes in and our skin absorbs, we have this sort of two-way street where cholesterol makes vitamin D, or the 7-dehydroxycholesterol, but also 7-dehydroxy or dehydrocholesterol is broken down into cholesterol. So it's a bi-directional pathway, and that's where we're going to see the activity of this, and that's how we end up with this whole relationship with calcium balance and vitamin D. And then, the VDRs, here we go. You see there's VDRs all over the place, and that's going to sort of affect everything. But the DHCR7 essentially is acting down here with bone mineralization, with this action on cortical bone. GC is actually similar to ApoE in the way that we determine risk. It's two RSIDs that come together. And then CYP2R1, this is another cytochrome pathway. And that has to do with vitamin D25 hydroxylase and hydroxylation in the liver. So you can see GC has activity all over the place. And this is a protein that is in the albumin family, whereas the cytochrome, as you know, cytochromes tend to be in the liver, and that's where it's going to be affecting vitamin D. And then as we move through these pathways, we're going to affect our intestinal bone and immune cells based on vitamin D status, because they will shift our vitamin D levels. So now, what does this mean? How do we use this? And I always really go back to basics. So I say KISS, right? Keep it simple, smarty. We always want to start with the basics. These are probably things that you guys have had hammered into your head, but they're really fundamentally important in terms of maximizing your patient's health and their genetic predispositions, right? So food hygiene, sit, side, Fletcherize, right? Make your food, shoot your food until it's a liquid. And I had a professor in medical school who would tell his patients, chew 31 times each bite, which is such a random number, but people would remember and utilize that. But sleep hygiene, we want routine, we want darkness, we want stress management, breathing, meditation. Where is your joy in life? Optimal elimination, you really want to be eliminating well in poop and sweat and respiration. Exercise, feed your mitochondria with movement. And I didn't talk about it with Ernestine Shepherd, but we really want to make sure that strength training is incorporated in aging. We've got solid research on strength training, supporting mitochondrial function, but also increasing bone density. And then minimizing burden by making sure air, water, nutrients, and friends are all a part of the life. And when I say air and water, healthy air and water. I mean, we have to think about where you live, whether you're in proximity to highways, do you need air filtration, do you have carpets in your home? Really got to take into account all of that. Diet and longevity. So these are just some studies on the people who've lived the longest. In Okinawa and both at Seventh-day Adventists are vegetarian. Okinawans have a highly fish-based diet and some meat, but a great deal of vegetables. And these are the folks that tend to live into their hundreds. So both of them have been highly researched to support their diet being an active factor in their ability to live longer, healthier lives, right? It doesn't really matter if you're living till you're 100 if you don't have any memory and can't get out of the chair. Well, I think so at least. So this is a nice little image on diet and longevity and the different places, the different dietary factors you can consider, how they're actually helping your patient. This is sort of a nice image to use with your patients to give them an idea of like, look, by increasing your fiber, you're supporting a healthy gut microbiome. You're shifting the metabolites, you're decreasing inflammation, and you're going to increase your longevity. And then overall, you just want to implement a strategy of optimizing methylation, looking at those SNPs and optimizing the nutrition, bypassing the pathways that are affected, balancing overall health by looking at these polymorphisms, and anticipating particular nutritional deficiencies, risk factors that are likely, looking at ApoE status, looking at bone health, looking at those cytochrome pathways, and supporting your patient through that.